So it's my pleasure to introduce Dr. Sylvain Costas. It's misspelled, I see Coates on your Zoom here. But Sylvain, I've known him for quite a while. During my NASA work, he's been running Gene Lab, and I can't say enough great things about him. So Sylvain got he has 2 master's degrees, one in physics and mathematics from Institut Polytechnique in France. And then he has an MS in nuclear engineering from Texas A&M, along with a PhD in nuclear engineering from Berkeley. And since I've known Sylvain, he's been the head of NASA's Gene Lab, where NASA's Gene Lab is essentially their omics data repository and analysis group. When I first started doing space science stuff, Gene Lab was a bit of an idea, but it was a little rudderless. And then since Sylvain's come in, it's amazing the transformation that it's gone from being just kind of an idea to a working group that does a lot of analyses, and they curate all these space scientists' data. They're awesome support and help for those that are trying to get into the omics and space studies. They've developed a working group of all these world-class researchers, multiple working groups of all these world-class researchers who come together. And Sylvain's been kind of the head of all this and has done an amazing job. I'm very in awe of the work that he's built up with Gene Lab. Also, correct me if I'm wrong, Sylvain, I haven't talked to you since this happened, but you're the head biological science lead at Ames now, I believe.
Yeah, I took over the other database, the ALSDA, so the non-omics database, and so it's basically all the open science database now that are space biology relevant are under my supervision, management, and you'll hear today also about artificial intelligence work that we now started as well.
So, wow. So because of COVID, I haven't seen you for a couple of years. So he's been
That's
promoted
right.
and taken on more responsibilities. So with that, I'll turn it over to you, Sylvain. Thank you for agreeing to come and talk to us. And
thank you, Joshua, for the introduction. You guys can hear me, right? And you can see my screen?
Yeah, we can.
Okay, good. Don't hesitate to talk to me because, like I said, presentation I've dropped before, and then I see my screen right now and I see nothing else. So I have no idea if I'm still online or not. So, okay. Thank you so much for the introduction. I prepared a lot of slides, and I know I have a French accent, so some of you may have a hard time understanding sometimes what I say. So I'll try to articulate, which is something difficult for a French person because we tend to speak with our mouth closed. So it's just a French thing. So anyway, hopefully I'll go slow enough. And if I don't have time to cover everything, that's okay. I'll try my best. Okay, let me start with a simple introductory slide about experiment done in space. You can think of the International Space Station as as a lab, a floating lab around the planet, and we're doing amazing things on the ISS these days. You can see how we grow in plants. We can do PCR. We can actually do sequencing with the MinION, which is kind of on the field type of a sequencer technique. And then we have all these amazing instruments that are becoming smaller and more powerful as time goes on. So, we are dealing with an exciting time in terms of generating data in space directly. And then in terms of the space biology, which is the division that funds Gene Lab and the Open Science Data Repository in general, let me give you a couple of important factors that we're very keen in understanding. We're really trying to understand how the space environment, maybe I should turn this off. We're going to see a bunch of messages. Okay. Space stressors, for instance, that we're interested in space are radiation exposure, altered gravity, harsh temperature and atmosphere, alters light cycles, isolation and closed environment, and dust and regolith that you would find on the moon and on Mars. So we're very keen on understanding the impact of all these different environmental stressors. And we really have been focused on omics in the past few years with GeneLab. So let me see if I can move to the next slide. Before I go into GeneLab, let me define open science in the context of NASA and our directorate that is called Science Mission Directorate. So I'm going to read this. We define open science as a collaborative culture enabled by technology that empowers the open sharing of data, information, and knowledge within the scientific community and the wider public to accelerate scientific research and understanding. This seems like a pretty common sense idea, but actually I have to confess that the biological community has been pretty slow at embracing open science. And one of the reasons for it is in the past, biological work has been done mainly by one PI and basically driving the effort. And so there's a sense of ownership, and it's really hard for our scientific community to share the information with others. And so this is a paradigm shift that needs to happen, and I hope the younger generation listening to this talk today can embrace it. It will give more value to your work if you share it. Yes, of course, when you share your data, you share the risk, or you run the risk of having someone else taking your data and telling a better story than you can. But at the same time, when we share data, we can put them in the context of a bigger picture. We provide all the information to let someone else reproduce work. And those are essential steps for science. We need to be able to reproduce people's work. So on a bigger scale, open science is the way to go. And we have a lot of challenges to deal with the ownership of your own work versus the benefit of the community at large. So let me describe quickly what are the open science resources here at NASA in the space biology community. On the right, you see 3 of them. So GeneLab collects and curates all the omics data. So that's proteomics, that's transcriptomics, gene expression, genomics, epigenetics, and so on. On the ALSDA side, which stands for NASA Ames Life Science Data Archive, we collect and curate all the phenotypic data. So it's microscopy, medical imaging on the animals. And then the bulk of the data we have at GeneLab and ALSDA are not astronaut data. They are animal experiment or plant experiment or microbe experiment or microbe collection in space. And then we have NBISC, which is a tissue collection. So it stands for NASA Biological Institutional Scientific Collection, NBISC. And NBISC is the companion to basically this database. If you see a data set that you like, you can actually see if there are tissue that have been collected and saved. And you can then request them and add more information or do more research on some of these tissues. So you can say, hey, they looked at the brain, but I am convinced the brain response I see here has a systemic effect that comes from the liver. So I want the liver tissue to show it. That's the kind of thinking we want you to have when you go into our database. So the 3 databases are extremely well connected, and we're building even more bridges between them to a point where actually ALSDA is now using the GeneLab engine. So actually you will be searching in GeneLab ALSDA. So there's one last program that is not a database, but that is really essential in this ecosystem is what we call the Biospecimen Sharing Program, BSP. And BSP is like whenever there's a mission in space station, like with animals, typically mice, there's a PI behind each grant that funds this research. So a PI will put a proposal about a study, and then they will fly the animals. But typically they're interested in a few tissues. They don't want the entire animal. So when the animal comes back and when we get all the tissue from the animals, there's a lot of leftovers, and there is no waste when we do an experiment in space. It's very expensive to fly mice, as you can imagine. So every tissue is collected and preserved in NBISC through the BSP program. And anyone can request it, having a good story, of course. So let me now focus on GeneLab. And one of the things that Josh mentioned about the transformation of GeneLab from being an idea to being a product, being something we can use. one thing that I noticed is when I joined NASA was that they were really focused on the raw data and the metadata, but there was really no afterthought on what to do with this data. So there was really old traditional archiving approach. And so the first thing we did is like, we need to turn those gigantic data sets into something meaningful for the majority of people. And we call that data democratization. So the way we've done that is we created new knowledge by transforming those raw data into something more meaningful. And we did that by establishing pipelines, bioinformatic pipelines that convert this data. This was done with the Analysis Working Group. AWG stands for AWG, and Joshua is is a member of some of our AWGs. I will talk about it in a minute. But those were essential. And on the left, you can see how we are democratizing and increasing the access of the information by, instead of giving data that only makes sense to data scientists, now a biologist and even a citizen scientist and a high school student can take this and understand what's going on. So this data democratization was really articulated around two important groups, the Education Working Group and the Analysis Working Group. So let me dive into the Analysis Working Group. So there's about 200 of people around the world, not only the US, this is international, 200 plus scientists getting together and focusing on one of these 4 topics you see here on the screen. So we have an animal group that really focuses on looking at all the different things from mice to C. elegans to Drosophila and other smaller entities. We have the multi-omics group, which tend to try to put things together. And sorry, I was checking my phone to make sure I was still talking to someone. So yeah, multi-omics group, under 208 people. And then this is really more the group that focuses on putting the different omics and different organisms together for a bigger story. The plant group focuses on plant, the microbe on microbes. All gene lab. There is a 5th AWG that's not highlighted here. I should have done that. I forgot. Is the ALSDA group. The non-omics group is now having its own AWG. And actually that AWG alone is 150 people. It's because we're covering so many different type of data that we need to bring a lot of different stakeholders. So we're working on this. Please contact us if you want to join it. We welcome student as well as PI. So, there's a link at the very top, genelab.nasa.gov/AWG/join. Okay, and on the education side, we have the EWG, the Education Working Group, and there are different activities that we have. The 2 big ones are Gene Lab for High School, which is a 4-week intensive training summer program, where we take high schoolers and we train them on bioinformatics. We use Galaxy as a platform to teach. Gene Lab for University is a boot camp training of 1 full week, 8 hours a day. And we put the information online. So any university can take our tutorials, can take our script and teach bioinformatics for space biology. So we encourage you to use the material. We also teach it ourselves, but only once a year. And so there's more information here about the course materials, and we're trying to expand this to all universities. So this is really something, if you have a program in genomics or a program in bioinformatics, I encourage you to use it. San Jose State University is now using the entire program that we put together. And actually they were our pilot. They helped us build this. And then finally, SLSTP is the undergraduate students internship that we have in the summer. Some of you interested in spending a summer in NASA, this is the link to click on. Okay, so Gene Lab is not a database anymore. Every time somebody is like, oh, you're an archive, it's like, no, we are an enterprise because we really have all these different layers. So we have the application layer, we have a data repository, we have a collaborative workspace, just like a Dropbox, but for science, where you can put your data, share your data with others, and so on and so forth. We have an environmental data portal. On the analytical side, we have an analysis platform that is Galaxy, as well now high-end computing clusters using Jupyter Lab for script. Sorry. We have a visualization platform. I will walk through you some of this. And finally, we have an API, which is a programmatic access point, so that you can, from your computer, from your command line, connect to us and start putting our data and working with it. Great when you're on the cloud. One thing we pride ourselves to be is FAIR. It's on the very left here. It stands for findable, accessible, interoperable, and reusable. Those are the acronyms for all the, any database these days needs to be FAIR. And so we built GeneLab around FAIR. We were lucky. We built it late compared to NIH. So I would say we're one of the fairest database in the world in terms of medical data and biological data. That's as simple as that. And again, the reason is that we're a fairly recent one. So we benefit from all the things that our pioneers have done in the field. So I would really encourage you to read about FAIR if you're not familiar with it. It's really important. Something unique to us too is on the source side, we have a team that generates data. So one thing that distinguishes us from NIH is that, remember the BSP program? We get some of this tissue. We at GeneLab can request it, and typically the analysis working group can voice their opinion on, hey, we really need more liver data in this database. Actually, we have tons of liver data. I should give another example. We need a lot of skin data in this database. Okay, then we can go and request the skin tissue, run omics on them, and put them online. The beauty is that we're not PIs. So when our team produces the data, we don't touch them. We actually unauthorized to touch them until they're publicly available. And that's really, we try to lead by example. This is the way you do open science. If you're really secure about your open science, you should have no problem as a PI sharing information before you even worked on it. I know it sounds really radical, but that's kind of like the true belief behind open science. And then the idea is that people will join you and work with you, and that's what the analysis working group are doing. So that's really unique to us. And the AWG love the GeneLab generated data because they're very, very clean and very well annotated. For time's sake, I will not dive into all these different points. Okay, GeneLab in a few numbers, and that's already outdated. It grows every month, but we have more than 350 studies. We've been growing very nicely over the years. We take every asset type in the omics world, but you can see actually how transcriptomic makes 69% of our data, and microarray and RNA-seq are equally represented right now. Having said that, no one does microarray these days, so expect to see in the next 10 years RNA sequencing taking over that bar and making the bulk of our data. In terms of what we look at, about half is space flight, sample coming from the space station or from satellite or from the space shuttle, if we look at archive tissues. But having said that, there are other things that we call them analog experiments, things done on the ground that try to mimic what's happening in space. So you can go to Brookhaven National Lab and expose samples to the accelerator to simulate space radiation. We have a lot of this data in our database. Or you can have mice being put with a little harness, and so they barely touch the ground, and so you can simulate the gravity on the moon or on Mars, so you can actually literally hold them up by the tails and have their hind limb unloaded from the ground, and then we can simulate all the back of the animals is in microgravity. So those are called analog experiments. On the human side, we have bed rest study, which is when we ask a person to stay in bed for 30 days or 60 days max, and we look at all the bone loss and all the changes that's happening, and we have omics data for these people as well. The goal here is to, in the FAIR principle, R stands for reusability, and that's really important. Reusing of data is important. I would say there's another R that people don't talk about is reproducibility, which is, good metadata, good description of the experiment, so someone else can repeat your work. So with this concept in mind, over the past 3 years, there's 38 papers that came out of GeneLab in some fashion, with 10 of them that were produced directly by our analysis working group. So kudos to this group of people. And the big splash was in 2020, we had 29 papers in Cell Press. Some of them made it to the Cell paper itself, the Cell, Cell, the journal. So, which is, as you probably know, I would say the top journal in our field. So super exciting about this. I encourage, if you want to know more about space bio, go check it out. There's a whole, it's called Cell, the Biology of Spaceflight, and look it up. If you type this on your computer, you'll find the special release. Okay, so let's go back. Where are we right now? What we've been building since then is this merging of ALSDA, our non-omics, with the omics database. And now we have a common API. And then we also have a common submission portal. So principal investigator now can go to one place at NASA to turn in their data. The omics goes to GenLab, the non-omics goes to ALSDA, the metadata are shared. And by harmonizing our data, our goal is really to, look at, we want to deploy, and I'm going to talk a bit about that, analytical tools to really analyze this heterogeneous data set in the context of the environmental information we had for a given experiment. So you had mice in space. Let's say there was a solar storm, and well, did that impact our animals? There was a high level of carbon dioxide on the space station for 3 days. Did that have an impact? So that's the kind of environmental factors we can look at, or they can be factors that are deliberately put into the system from the experimental design of the PI. All this is in the metadata, and we're using AI ML technique in addition to system biology. So that's something more internal that we're doing now to create a modeling portal. We're linking this data to other databases to infer information. One issue with space is that we have few data, so this external database can give us a context of what those data mean. And then our goal is to really create a predictor of, you can imagine a way to basically predict what will happen to someone when they go into space under a given stress. Let's see how I'm doing with time. Okay. So, and then we have a visualization tool that also helps you look at this modeling data. This one is not out yet. So it's coming up at the end of the fiscal year. So this is what we have right now in terms of Viz. This is the visualization platform. You can go to GenLab, go to visualization, and you can see here, I'm pulling one data set. Right now, we can do one single data set in GenLab, but the multiple data set is going to be coming out very, very soon. So soon you'll be able to look at multiple data sets together. In this example, it's an experiment done on lymphoblastoid cells, the human cells exposed to low level of radiation. And you can see here with the PCA plot, which is a principal component analysis. So if you take the gene expression from each of these group samples, there's a dot here on the PCA plot. And then if I zoom on it, you can see it's interesting how we see a very nice progression in this hyperplane space, where you can see you start with 1 centigrade in blue, 5 centigrade, 7.5, and 10. This is a very nice progression in this space with the PCA plot in terms of how that exposure to radiation move you in terms of the gene expression pattern and how it distinguishes you from the other. So PCA plots are kind of the classic first thing you look at when you look at omics. It's pretty much, that's the first thing I look at typically. Okay. On the ALSDA side, which is the non-omics, we are building this as well. So hopefully we'll have something, but here it's an example of multiple data sets. So we're looking at a bunch of different missions, either on the space shuttle, on Bion, which is a satellite, or on SpaceX, so ISS, and the duration of the mission. And here you're looking at flight versus ground control. So those animals in flight, and then we look at the ratio. And then here, these are typically a typical ratio of bone density. So you can see how overall across all these different missions, you have a lower level of bone density in space versus ground. And that's the quantification here. You can also look at trabecular thickness. And again, you see this significant drop in the thickness of the bone. So this is the power of putting things together. You can reuse it and you can look at it in a bigger context. Okay, quick reminder of what is space radiation. Space radiation, we call it galactic cosmic rays, GCR. 87% is proton, alpha, well, helium, alpha, 12%, and then high Z, high energy, high Z particle, 1%. And then if you, so if you are on the ISS, you've got 50% GCR. So sorry, ISS and LEO are pretty much the same. It stands for low Earth orbit. So when you're on the ISS, 50% is this GCR, and then 50% is trapped protons that are circulating. You check it out. It's called South Atlantic Anomaly, SAA. and that there are regions where there's more trapped protons, and as the ISS circulates, it gets exposed regularly. Moon and Mars is a bit different. We have a lot of secondary neutrons from the impact of the particles on the moon. And the beauty is that I'm sorry, the little cartoon you saw at the end is like we can simulate this space radiation at Brookhaven National Lab, which is on Long Island, New York. One thing of interest here is that's the environmental portal for GeneLab and ALSDA. I should remove the GeneLab logo because now this portal lives for both ALSDA and GeneLab. We built it at GeneLab, but it really belongs to the bigger community. And what you see here is that the dose rate measured by the instrumentation on the ISS changes as a function of when you fly, 2014, 16, 19. And basically, what happened is when you have solar minimum, you have the highest dose. So that sounds a bit contradictory, right? The solar minimum is when you have very few solar wind, you're not pushing out the particle coming from deep space, and so they come and they hit you harder. So you have this higher dose of radiation from the GCR at solar minimum. So you would say being at solar minimum is not a good thing because of that. But the good thing about solar minimum is that you are less likely to have a solar particle event, which are more likely when you have a high solar wind. So pick your battle. You can either get a huge dose in one shot through the sun through this solar particle event, but you have a lower average dose rate from the particles in space, or vice versa. And so this is a kind of metadata you want to capture when you look at our data, because you need to look at them in the context of the radiation. That's one example of the influence of the environmental data, and we can look at that. So we built this. You can go there and play with it. It's really fun to visit the data we have collected so far. I think I'm going very slowly. In the context of radiation and study of radiation, 10 centigrade is what we consider the low dose. So for those of you who don't know that, above 10 centigrade, if I give an acute dose of 10 centigrade, I have a significant increase of inducing cancer in a person. And the reason we know that is because of the atomic bomb data survivor. The survivors of the A-bomb, Hiroshima and Nagasaki, have been studied for 80, 90 years. I don't know, maybe not that much. Let's say 60 years. And the thing of interest here is that where the bomb was dropped, we know exactly based on your location, the dose you receive, very precisely actually. And so we've been monitoring these people. And so we have a very good idea of when radiation becomes dangerous in terms of cancer. There's a lot of discussion around this topic because what we've done is we took this data and we extrapolated them back all the way to very low dose. But below 10 centigrade, the statistics are not there. We can't conclude that there is a risk below 10 centigrade, at least not through this process. And that's been a big issue because all the regulatory agencies are assuming what we call LNT, linear no threshold. That means that no matter how low the radiation is, it's dangerous to you. And there's many school of thought that thinks that you can't extrapolate down. It's kind of like saying, I give you 10 ibuprofen, you will die of a liver failure. I think that's something like this, so maybe 20. Therefore, ibuprofen is bad for you. And if you extrapolate down in the dose, you can estimate the risk. It doesn't work this way, as we know biology is a nonlinear system. And so there's been a lot of debate. And the problem is in space, we are only exposed to chronic dose. So we're really in this low dose regimen all the time. So it's an important question that remains to be solved. So plenty of work there to do. Okay, so here I'm going to jump to my personal lab. So I'm a project manager, but I'm also a principal investigator. And I've been looking at animals and humans. So I use human blood and mouse skin. And together I'm trying to look at DNA damage. And I'm trying to correlate what we see in the animals to humans. So I only give you a snapshot of what we've done. There's a bunch of papers we published over the past years, and all the human work is basically currently in being prepared. So I can't share too much of this data yet. So I'm going to focus on the human data. So the first thing we notice is that this nonlinear response of if so sorry, let me go quickly on this. I want to show you what it looks like. So you see these images at the bottom. Those are mouse skin cells. The 0 gray is what they look like without radiation, and then 1 and 4 gray shows you with X-ray what they look like 4 hours after you expose them. And those little dots, they are DNA damage. And so we're using something called p53 binding 1 protein to label it using immunohistochemistry and microscopy. And we do high throughput microscopy. And those graphs on the left represent millions of cells quantified across 15 different strands of mice. So we use 15 strands. Each color represents a different strand. Each strand is made of 6 animals, 3 males, 3 females, and only their skin. So we clip their ears and we grow the ears in a dish. And when we have enough of it, we expose it to X-ray and gamma ray. And to make a long story short, what we notice is that as you increase the dose, you see a nonlinear response. And it's actually shown here on the bottom right. As I increase the dose, I know I'm inducing more DNA double-strand breaks, which is what we're labeling here, DNA double-strand breaks. But the biology is a bit more complicated than that because I'm not using a marker of DNA double-strand breaks. I'm using a protein labeling the DNA double-strand breaks. And what our data suggests is actually, as you increase the dose, you see a similar number of foci, those little dots, but they are larger and brighter. So our interpretation has been that many DSB come together into one repair centers, and that's a concept that my lab is pushing for. It's still being debated, and some people don't believe in it. So full disclaimer here, but basically the concept is that you have this repair unit that comes together and fix the DSB. And the hypothesis of my lab has been that this is a bad thing to do for a cell because you run the risk of, you basically run the risk of having a bad repair, or you can mix multiple DNA double-strand breaks together and you can have a rearrangement of the chromosomal level that could lead to cancer. So there's been a hypothesis in my lab that this process of putting DSB together for repairing is something that is not a good process for the cell. And again, you have to remember that on Earth, this kind of dose in natural evolution, we never got exposed to that kind of dose. Those are 1 gray and 4 gray. Those are clinical dose for treatment of cancer. They're really big. 4 gray to a human will kill you. Full 4 gray on a person kills you. So those are very, very high doses. And the thing that's interesting here is that, for the particles in space, we see a lot of this kind of high energy deposited. So there's really a lot of question about how this kind of repair process could really be a problem in space. On the DNA damage, we show that those DNA damage can predict radiosensitivity. So we did blood draw and finger prick both, and we took the blood and exposed it to this simulated space radiation. We also had a partner that actually was at the hospital and took the same blood draw of some of these people that were actually being treated for prostate cancer. So those were male. And we could follow their outcome in terms of their sensitivity to radiation. And basically that cartoon summarized this paper in Cell Report, where we show that if you have a low baseline, you tend to have a stronger DNA damage response through the markers and a lower toxicity, which could confuse people. We see a stronger DNA damage response, but we see a lower toxicity. Basically what it tells you is that your body is responding correctly and is reacting to the radiation and doing something about it. And what we notice is people who have a very high baseline level of DNA damage tend to not respond much to the radiation. And on the long run, they basically seem to be more having more toxicity. So let me walk you through some of this data. So those little DNA damage I'm just showing you, they're interesting. If I take a blood draw of, this is about 300 or 400 people, you can see that as you get older, you have more and more DNA damage. There's no difference between male and female. There is those DNA damage markers also seems to correlate with infection. So if you have a CMV infection, you have a higher background. If you do finger prick, this is a bit cleaner assay. We saw a stronger edge dependence. And then finally here on the bottom right, what we're showing is that each assay has different baseline level. So the best is when you take the finger prick, we take the blood and we immediately fix it and do the analysis. So it's what gives us the lowest background. And I think this background is the true background that you would see in the body. This one is a baseline that we do, those blood draw, what we have to do is we got to take the white cells out of the blood first, and that's done on live cells. So by the time you extract the PBMCs, you end up having induced some stress, and you can see it here that it's adding a higher level of DNA damage. And the worst is once we freeze the cells and take them to Brookhaven to run the experiment and thaw them, their baseline is even higher. It makes total sense. And the point of this graph tells you that when you do an experiment, make sure you use the right control. If I do an experiment at Brookhaven, I want to look at that baseline, not that baseline. It would be really a mess. Okay. And then here, this is what I was telling you. If you're a non-reactive person at the clinic, the same blood of these people have a strong response with those. And then if you're reactive, you have a low response. Okay, same thing with the baseline. Lower baseline, good response. It's the same data shown differently, but the point here is that if you're non-reactive, again, even during mid-treatment, you have a higher response of DNA damage. Okay, here is something that was really surprising to me is that, right now we're looking at DNA damage and a response to toxicity, but we decided to look at cytokines, which are, other type of markers in the blood. And what we observed here is that actually a patient that had a low, actually, I'm sorry, I'm switching from patient to donors. Those are healthy donors. So we had a cohort of like 400 donors. We took the 12 donors that had the lowest baseline for DNA damage and the 12 donors that had the highest baseline for DNA damage. Then we expose that blood to radiation and we look at cytokine response. And the thing that's really remarkable is that the high dose response, the people that have a low baseline also show a very strong cytokine response. So basically, that baseline tells you that your body is responding. You are responding to the stress. These folks seem to not respond at all. Okay. Now we're moving to the human cohort. I can't show you the data, but basically we're doing a genetic association data between this kind of biomarkers, DNA response, and the genetic of these folks. So stay tuned. This work is coming in probably a year. We're done collecting the data. We now have to do the analysis. It takes a while. It's pretty heavy. Okay, let's see. Where am I with time? Do you guys want me to leave space for questions?
Yeah, that would be great. Maybe, I don't know, another 10 minutes.
Okay, we'll get where we get. It's okay. I think I'm 2/3 there. And these are lighter. So let's back up now and let's think about the bigger picture. What are the challenges in space biology? And then here I'm going to focus on machine learning. The first challenge we have is like I mentioned, low sample size, high dimensionality. That's expressed here. I have 30 samples and I've got 50,000 genes. Well, that's a bit of a problem for machine learning techniques. You're going to overfit everything you do. The other problem is we have a bunch of different type of data. We call them heterogeneous data, and we have sparse data, meaning that we don't always have all the same data. Sometimes we have them, sometimes we don't have them. So there's sparse metrics. And finally, we typically operate in what we call out of distribution situation, where we describe a response for one type of statistical distribution. And then when we move to other kind of questions, suddenly everything looks different. And the statistics that we apply or that we assume may not necessarily fit. And so those are the kind of challenges we have in space. Okay, so we have FDL, which is a Frontier Development Lab. Every year has a challenge, and one challenge for NASA. So they cover heliophysics, planetary, and then there is one section dedicated to space health. And then this was the group of last year, and they use a tool called CRISP. So let me walk you through this. It's pretty cool. So the challenge here was to give them 3 human data sets and 3 mouse data sets. The first thing they did is they merged the data set because obviously we have a lot of commonality with mice, but we're not mice. And so we had to use genome homolog mapping to take the mouse gene data and turn them into gene that we know in the human side. And so then we created this combined data set. And then we ran it through something called CRISP. So CRISP is a causal inference model in machine learning. So we can take any kind of data, microbiome, epigenetic, genetic. So in our case, it was transcriptomic, and also some, I think it was mainly transcriptomic. And then the first thing you do aggregation of the features, so you reduce the dimensionality of the data. And then we use an ensemble of AI ML technique on the causal inference side. So these were 5 different causal inference methods. And we vote for what is the one thing that all these methods sees being true. So, and the goal is to identify the cause to an outcome. So that, I know I'm very abstract, so I'm trying to be a bit more specific. I said I had 3 mouse data set and 3 human data set. And you can think of this as a target. Your target is your radiation state. So are you not exposed to radiation? That's going to be your target. You can plug this in as a value. You either 1 or 0. Okay. And now what you can do is you can optimize the features that are causal in this network and find what are the most common network for all of these. And you do this with this different causal method. And then you do a voting where you keep the network that are being identified as the most likely. And doing this, we identified top 20 genes that were causal for the radiation status, either you irradiated or you're not irradiated. And what was really encouraging in this exercise is that 4 of these top 20 genes were cancer genes, so genes that are known to be involved in cancer. And as I said, radiation can induce cancer, so it's interesting to see this signaling being picked up by AI/ML. Again, the beauty of this, there is no pathway, there is no pre-knowledge. It's literally just using this kind of technique in an unbiased manner. The other type of thing we do is called knowledge graph. So basically here you can imagine how you have a genes being linked to a disease, to a cellular component. And so Spoke is a tool at UCSF, and our collaborators, Sergio Baranzini, who is the PI, let us use this tool. And it was really fun because we could look at the same data in a different angle. So let me try to explain something. If we're going to focus on gene and anatomy, if I start looking at the gene expression with folding in the anatomy, what can I do? I know I'm very abstract, but hopefully the example will make sense. So here we look at thymus, liver, and spleen. 1, 2, 3, I think it was something like 9 different data sets. That was in GeneLab. And you can see 3 different tissues. And when you do a PCA plot first and you look at how those changes in space are, so that's what you can see here. Sorry, before I even talk about changes in space, those are the full data sets. So it's flight and ground control mixed together. And you can see that the PCA plot shows you that basically each study cluster on its own. Makes sense. Within each study, that's the second plot, if you color code those points for what was on the ground, which is basal and ground control, and what was in space, which is blue, they don't separate very well, maybe a little bit. And actually, if you do a gene expression difference between flight and ground, you see very little overlap between each tissue, thymus, liver, and spleen, almost no genes overlap, very frustrating. So it's really hard to give a bigger picture with this. Now, here is the crux of the matter. You repeat this, you give the gene expression as a vector, and for time's sake, I'm not going to go into how those vectors are generated in Spoke. But basically, they represent one node, is the gene expressions. Remember the gene node. But there is another node, which is the anatomy, and those genes are together. So if you can, in a nutshell, the fact of telling I'm thymus and this gene's expression comes from the thymus, I can now deconvolve the bias brought in by the tissue, if I try to summarize things. And if I look at the symptom nodes, which is the beauty of this graph knowledge, is now I can link these genes to a symptom node, which is yet another database that I can pull from it. Here is what I get. Space motion sickness, which is kind of crazy, hearing loss, and conductive. Those 2 phenomena have been, space motion sickness is the obvious one. And then you see here ground versus baseline, okay? So space versus baseline and space versus ground are those ones. They separate very nicely. What it tells you is that basically if you're in space, those animals are probably motion sick, something you can't really quantify because they don't really speak. and it's not like they're throwing up or anything like this. So, the animals are not feeling right, but the molecular signature is picked up. And the beauty is that when you look at the anatomical node that picked that up, you see a bunch of brain, head, midbrain, pituitary, pretty much a bunch of anatomy node that makes sense for this kind of motion sickness. But remember, I got this using thymus, liver, and so it's the beauty of this. We are capturing the systemic response in this tissue by deconvolving. So very powerful technique. Very excited about it. I will finish on this slide. We are working right now with the AI group to generate a bunch of different approaches. We have something called the benchmark data set, which are going to be large data set. There's going to be a DNA damage data set, a bone loss data set, and what's the third one? And a transcriptomic data set for people to use and train machine learning on it. And that's going to be space versus ground, or radiation versus no radiation. So stay tuned. I'm going to stop there. I'm going to go quickly to my concluding slide or acknowledgement slide. This is the Gene Lab team over the years. This is the AI team, and this is my lab team. And with